Welcome to my next class on biotechnology. In previous class, we were discussing about agricultural application of biotechnology in agriculture. Similarly, the next topic, as per your syllabus, what we have is application of biotechnology in medicine, in medical field. Application of biotechnology in medical field. These are the two very important arenas in which biotechnology has contributed a lot. One is agriculture, that take care of the food production, then animals, then it take care of horticulture and agriculture crops. Then apart from that, the next very important requirement for human being, for the life, for the sustenance of life, for the continuation of life is health. So the next very important application is application of biotechnology in medicine or medical field. It has played a pivotal role in these two fields, and apart from these two major fields, biotechnology has polished each and every corner, each and every arena, each and every field of life, and as well as the science. So these two major fields we have to discuss as per your syllabus is concerned. In previous thing we have discussed about the application of biotechnology in the field of agriculture. There we have seen transgenic plants. So here in this, which is the topic today, application of biotechnology in medicine. So as far as this major topic is concerned, two subtopics we have discussed. One is about the insulin, production of human insulin, production of human insulin is combined to form humulin, one application. The second application we have to discuss is about gene therapy, very interesting topic, right? Gene therapy. So these two major topics, there are again certain other subtopics are there as far as the application of biotechnology in medicine are concerned, but according to your syllabus, these two very important subtopics we have discussed, that is the production of human insulin, and then we have to discuss about gene therapy. After that, very few small topics are there in this chapter. It may take around 3 to 4 classes to complete that, to those small topics, important topics. So now, before we go into the production of humulin, we have to go for introduction part. So as far as genetic engineering is concerned, or biotechnology is concerned, or recombinant DNA technology is concerned, you might have studied in your 11th chapter about recombinant DNA technology, how to carry about recombinant DNA technology. At your level, this recombinant DNA technology can be also referred as genetic engineering, but when you go for research at higher level, these two changes a little bit, slight difference. But at present, we can consider genetic engineering or recombinant DNA technology are one and the same. So you might have studied those topics in detail in the previous chapter. So by recombinant DNA technology or by genetic engineering, so far more than 30 medicines or drugs have been prepared, have been designed, have been made through recombinant DNA technology. 30 to 35, more than 30 drugs, 30 medicines have been so far synthesized with the help of genetic engineering, with the help of recombinant DNA technology. Among them, more than 12 drugs, medicines are available in our country. Right? So far, more than 30 drugs, medicines have been prepared by using genetic engineering, by using recombinant DNA technology. Out of those 30 to 35 medicines in India, here there are about 12 medicines are available, and all are approved for human use. Some medicines are not for human, they are for other animals also, they have been designed. But so far, whatever they have been marketed in our country, around 12 or more than 12 medicines are for human beings. We can use recombinant DNA medicine, and we are getting vaccines also now. So medical vaccines, recombinant DNA vaccines, many more are there. So this is the introduction part of this application of biotechnology in medicine. The next topic, moving to our topic, is production of insulin. Before we go into actual process of production of insulin, we should understand what is insulin, what is the importance of insulin in our life. You might be knowing about diabetes mellitus we have discussed in the previous chapter also, that the last chapter of your first year we have discussed about that. So here, when we talk about insulin, you all know that insulin is a hormone basically, but basically every hormone, every protein, whatever that is being synthesized in the body, whatever is prepared in the body is a protein. That's why you have studied in molecular biology chapter as protein synthesis. So whatever they are produced from exocrine glands, enzymes, from endocrine gland, hormones, basically they are proteins. Later, as per the requirement of the body, based on their functions, based on their origin, we consider them, we categorize them into vitamins, into enzymes, into hormones, into proteins, into molecules, like that. So here, you all know that insulin is very important. It is produced, synthesized in our pancreas. You also know that pancreas is a mixed gland. What gland? Mixed gland. Fine. Clear. It's a heterocrine gland. What do you mean by heterocrine gland? One part of it acts as endocrine. That is, it secretes hormones. The other part of that same hormone acts as exocrine gland. That is, it secretes enzymes. We have seen pancreas is a mixed gland, compound gland, heterocrine gland, because one part acts as exocrine that produces enzymes like pancreatic lipase, fine, and then pancreatic amylase. The other part of it secretes, acts as an endocrine gland. So the gland that secretes hormones, you all know that hormones are chemical messengers, enzymes are biocatalysts. So here this endocrine part of pancreas is made up of 5 types of cells, but at your PU level we discussed only 3 types of cells: alpha, beta and gamma cells. Fine. Alpha cells secretes glucagon, beta cell secretes insulin. That's what is our topic today. Fine. And in the next class as well. So alpha cell secretes glucagon, beta cell secretes insulin, and gamma cells secretes somatostatin hormone that also acts as an anti-growth hormone. For your neat information, we will discuss about neat questions also. After 1 or 2 topics of this chapter, I will discuss about the neat questions, 5 questions in each class. So somatostatin, that is 3 cells. When you go to medicine, MBBS, there if you study about pancreas in the human physiology, we say that they teach you that there are 5 types of cells in pancreas. That detail you study over there. So here, one is alpha cells that secrete glucagon. Now glucagon is also a hormone. What is the function of glucagon? To increase the amount of sugar level in the blood or in the body. To increase the sugar level. So it increases the sugar level in the blood. Hence it is known as hyperglycemic factor, because it is increasing the glucose level. Hyperglycemia. Whereas the opposite function of that is exhibited by insulin. So what is the function of insulin? It reduces means what? It brings to normal. Whenever there is an excess of sugar in the blood, whenever there is a more amount of sugar is circulating in the blood, that is problematic. Right? It is not optimum. So it has to be brought to the optimum level. If it goes down, hypoglycemia, hyperglycemic factor glucagon has to show function to increase and bring to optimum. Now the next abnormality, hyperglycemia, when it has increased, right? So that's also problematic. Maximum, I have told many a times, maximum is also problematic, minimum also problematic, whether you consider blood pressure or sugar level, whatever it is. So what is required, what is desired, what we need is optimum conditions, right? So to maintain the optimum conditions, whenever there is increased glucose level in the blood, more amount of glucose is circulating in the blood, that more amount has to be reduced and brought to optimum level. So to decrease, insulin acts. When there is a less amount of glucose level circulating in the body, glucagon acts. I hope it's clear. Now, you also know about the disorders. Your grandparents, in your family members, in your parents, right? If they are suffering from sugar, what they call sugar complaint, sugar patient, right? Everybody has sugar, but we have optimum level, they have different levels. hyperglycemia, hypoglycemia. So most of the people, they suffer from diabetes mellitus. Not going into detail of diabetes mellitus because there are many types of diabetes mellitus. We have discussed also, one is juvenile diabetes, other is maturity onset diabetes mellitus, and again one more type is insulin dependent diabetes mellitus. The other is known as non-insulin dependent diabetes mellitus. I have discussed this in first year when we were talking about digestion chapter, when we discussed about there in case of pancreas. I have told you in detail. Now there is no platform here to go in detail and not required also as far as this chapter is concerned. Right? So when diabetes mellitus occurs, the amount of glucose level is high in your blood, insulin has to be produced. Now, one factor I will tell you now. In these patients, why glucose level increases? Because in their body, in their pancreas, from the beta cells, adequate amount, required amount is not produced. Required amount of insulin is not synthesized. Needed amount of insulin is not synthesized. Adequate amount of insulin is not produced by the body cells from the beta cells. Hence, external insulin has to be taken. So you might be knowing sugar patients will take tablets that help in the production of insulin hormone. Insulin hormone is also a protein, right? So when these tablets are taken directly, they used to take insulin injection. You might be knowing if it is more than 400, more than 450 immediately, directly insulin injection has to be taken to the body so that the amount of such a high glycemia should be reduced immediately because it damages other organs, right, in the body. So insulin injection is taken. Insulin injection is given to diabetic patients, sugar patients. So which is that insulin? So in our body it is not being produced. Now the question may arise in your mind, so why it is not being produced? There are many factors for not being synthesized in our body, the protein insulin. Why? Because sometimes in juvenile diabetes what happens, or in 8th chapter also we have discussed about autoimmune disorders. Due to autoimmune disorders, autoimmune disease, our own cells, our own antibodies kills, damages the beta cells, thereby these beta cells do not produce or cannot produce the adequate amount of insulin. That is one factor. Or in some people about 45 years of age, 50 years of age, insulin is being produced, but after production of the insulin in their body, what function has to be performed by the other organs? The function according to the order given. That's why chemical messenger, it gives a message, it gives a hormone is a messenger, hormone is a chemical messenger, it gives a message, it gives order to other organs to reduce the body sugar level. These organs will not accept to the orders or the chemical message brought by insulin. This is also one of the factors. Even after production of the insulin, adequate amount of insulin, the normal function in the body do not takes place. That is also known as the resistance against the insulin action. The concept is known as resistance against insulin action. So this is also one of the factors. So many factors are there. One is genetic factor, other is autoimmune disorder, one is the hereditary factor, because of tensions, because of inadequate sleep, because of tensions, because of problems. Hormones do not produce in the body normally, hormones do not function in the body normally. So many reasons are there for that. So to overcome these problems, right, clear, we have to take the external insulin. So far in our country or other also, whichever the insulin the patients were being given injected is that insulin that is extracted from other animals like cows, buffaloes and also pigs. So here the problem is the insulin that is produced in cattle and in other animals, that is specific to that particular animal. You got my point? Human insulin is slightly different, fine. And the animal, in the cow, the insulin that is produced is specific to cow, specific to bulls, specific to other animals. Clear? Fine. So that specificity is there. Human insulin is slightly superior, slightly purified, more better, more specific in action, in structure as well to some extent. So because of this problem, whenever the external insulin, the insulin that is obtained from other animals when injected into human bodies, into the patients, they used to show certain side effects, certain allergic reactions, exaggerated reactions, allergic reactions, abnormalities, certain problems, right? So to overcome these problems and to make the humans insulin, we have to understand this. Before we go into the production of human insulin as topic, the introductory part is we have to understand the structure of insulin. Whenever in our body, from the pancreas, when beta cells produces insulin, this insulin will always be a pro-insulin. What we call it as pro-insulin, because what do you mean by pro here? Pro refers to inactive. This pro-insulin is what is nothing but inactive insulin. When we say inactive, it means it is non-functional. It is not in the form of performing a function. Non-functional. Why? Because pro-insulin consists of 86 amino acids. 86 amino acids. I told you, insulin is a hormone. Every hormone is nothing but a protein. That's why in our body, protein synthesis, what we call as protein synthesis takes place. Hormone is a protein, vitamin is a protein, basically enzyme is a protein. So here, whenever pro-insulin, it is inactive. This pro, because of this pro factor, this is inactive. Whenever it is produced in our body, it is in inactive form, pro-insulin form, non-functional form. So why? Because whenever there is a hyperglycemia, whenever there is amount of glucose increase in the blood sugar level, this pro-insulin will be converted to insulin. So how is it possible? So now this insulin is what? It's active insulin. What insulin? Active. Now what do you mean by active? It is functional. It is functional. So now this amount, whenever beta cells secretes insulin, this insulin is produced in the form of pro-insulin, which is inactive, non-functional, because it has 86 amino acids. We have to understand the structure of insulin before we understand the production of human insulin by biotechnology or by genetic engineering concept. This is the base to understand. This is the next topic. So now here, this pro-insulin has 3 chains. One is known as alpha chain. What chain? Alpha chain. Other is known as beta chain. And the next one is known as the C chain. C chain. Alpha chain consists of 21 amino acids. Beta chain consists of 30 amino acids. Then C chain consists of 35 amino acids. I told you it's a protein, and you all very well know that every protein is made up of molecules called amino acids. Amino acids are the building blocks of proteins. Alpha chain is acidic in nature. Remember, A for alpha. Acidic in nature. Beta chain, basic in nature. What do you mean by that? Alkaline in nature. This chain is acidic. Its pH is acidic, its value is acidic, pH value is acidic, it's acidic in nature. Alpha chain having 21 amino acids. Beta chain, 30 amino acids, it is alkaline in nature or basic in nature. You can easily remember A for alpha, acidic chain, B for beta, B for basic chain. Now you can guess C chain, it's neutral in nature. Right? One is acidic, alpha chain, the other is basic, beta chain, the third chain is a neutral chain. Alpha chain consists of 21 amino acids. Beta chain consists of 30 amino acids, C chain consists of 35. It is also in increasing order. 21, 30, 35. Easy to remember. I hope it's clear, the formula. Now, this constitutes about 86 amino acids. We satisfy that factor now here. Now, I told you that it is an inactive insulin. Why inactive? Because these many amino acids are there. So here, C chain will be removed. What will be removed? C chain having 35 amino acids. We don't have an option here to tell you those amino acids. Remember, and that much detail is not there in PU level also. It starts from alanine, cysteine, fine, and then phenylalanine, serine, methionine, all those series are there. 21 amino acids like this. 21 amino acids are there. Then 30 amino acids are there. Then here 35 amino acids are there. This is alpha chain, beta chain, and C chain. Yes. Now here 21 amino acids. Then here 30 amino acids. Here 35 amino acids. Clear? So these chains are interlinked by disulfide bonds. Disulfide bonds. These chains are interlinked by disulfide bonds. Clear? So where they are connected, wherever there is cysteine amino acid, cysteine is there. In alpha chain, 7th position cysteine is there. 7th position here, 11th position beta chain. In alpha chain, 19th position. in beta chain 20th position. So they are interlinked, they are cross-linked. 7th position alanine, serine, cysteine, methionine. When it comes to cysteine, 7th position alpha, they are connected to each other. This much detailed structure is not there, but to understand, this is the molecule of insulin. 7th position you have cysteine. Here also 11th position you have cysteine. Here 19th position you have cysteine. Here 20th position you have cysteine. So at the cysteine level, these two chains are interlinked by disulfide bonds. What bonds? Disulfide bonds. So this is the whole structure of insulin. Insulin is a hormone, protein, amino acid. So at the cysteine amino acids specificity, this is what I told you. Human insulin is specific to human beings. It is slightly different from animal insulin. So cysteine, at the cysteine structure, 7th position, it is connected by disulfide bonds to beta chain. Similarly, beta chain is connected to C chain. Now, whenever this is there, it is inactive form. So whenever it is required in the body to perform its function, so from inactive insulin, that is pro-insulin, has to be converted to active insulin. So what is there here? Carboxypeptidase, one enzyme, carboxypeptidase. Carboxy, carbon atoms and all those things in chemistry we have studied. Peptidase proteins, peptidase, ACE enzyme, the enzyme that splits proteins, that digests proteins, peptidase. Carboxypeptidase is an enzyme, fine, that is being secreted again by pancreas only. See the nature, how smart it is, how beautiful mechanism is here, right? Or alpha trypsin also we can use. I will tell you that in the next topic. Carboxypeptidase enzyme is again secreted from the pancreas, from the exocrine part. So this enzyme splits, it removes the C chain, it breaks this C chain, it splits the C chain, right? When inactive protein is acted by an enzyme known as carboxypeptidase, it can be asked in your NEET also, right? Remember that point. So this enzyme acts on this protein, acts on this hormone, pro-insulin or inactive protein. When it acts, this C chain is removed, dissolved, digested. These amino acids are again recycled. That's all the different topic again. So here, so this now 21 plus 30, how much amino acids? 51 amino acids. When the insulin becomes from 86 amino acids, it is being reduced to these only two chains, alpha chain and beta chain, having 20 amino acids, 21 amino acids and 30 amino acids, 51 amino acids. Now this insulin is what insulin? Functional insulin, active insulin. It becomes functional in our body, it becomes active in our body. So here, this is how the structure of insulin. It is very much required to understand the structure of insulin. So in our next class, we will discuss about the production of human insulin. Do take very good care of yourself, right? God bless you, good luck, take care.